Hello students, and we are back. This is Dr. D. Today we are going to continue with endocrine system, talk about steroid versus non-steroid hormones. Alright, so in our last presentation we had said steroid hormones are all derived from lipids. Alright, so here the basic structure is like that of a cholesterol. So they are all derived from cholesterol. Those of you or those people who say all cholesterol is bad, that's not true at all, because there are certain steroid hormones in your body that originate or are developed from cholesterol. Non-steroid hormones are protein in nature, glycoprotein in nature, peptide or amine. Keep one thing in mind, steroid hormones, because they have a lipid base, they're not soluble in water, and of course non-steroid hormones will be very much soluble in water. Alright, how do steroid hormones work? So here what they do is they are going to diffuse through the cell membrane, but always remember, for once it diffuses, well, how does it diffuse? It will diffuse because what is their composition? What are they? They are lipids in nature, right? And what's the cell membrane? A cell membrane is a phospholipid in nature. Now, if you have ever played with two drops of oil coming close to each other, if you bring two drops of oil close to each other, what will happen? They will merge, they will fuse, right? Same thing. A hormone, a steroid hormone will come and will fuse with the cell membrane. Once it fuses, then it's pinched off inside the cell membrane and it'll bind to a protein receptor inside the cell. All right. So here what happens is where it binds, it is transported to the what? To again the nuclear membrane. The nuclear membrane also is a phospholipid where it will detach from the carrier protein and will enter the nucleus. All right. Once this is so, then what happens is a hormone based or hormone receptor will bind again to the steroid hormone, and these two will then be directed to bind at a particular site of the DNA chromosome. All right. So once it binds the DNA, either it'll turn the gene on or it'll turn the gene off. Well, what is on and off? If his gene is on, then the DNA is going to produce mRNA and is going to produce protein. Off means that now it is not going to be, the DNA is not going to produce mRNA and therefore no longer it will produce a protein. Got it? So it will either turn the gene on or off. It will produce something or not produce something. And like I said, turning a gene on, messenger RNA is transcribed, transcription takes place. If on the ribosome, then the mRNA is translated to a protein. So a protein is produced or not produced. Okay. For a non-steroid, of course, it cannot diffuse across the cell membrane. Why? Because the cell membrane is made up of phospholipid, right? Here what happens is it is not going to be able to diffuse across like a steroid hormone. Instead, it binds to a glycoprotein receptor, all right, on the cell surface on the plasma membrane. A non-steroid hormone, therefore, is called the first messenger. Important. So, first messenger binds to the receptor. All right. Now, as soon as it binds, it will now activate an enzyme inside. What's that enzyme called? Adenylate cyclase. Adenylate cyclase converts or helps to break down ATP into cyclic AMP. All right. Cyclic AMP. Sorry. I'm going to rewrite it out here. Cyclic AMP. Now, cyclic AMP, what happens is called the second messenger. So, the first messenger is the hormone. The second messenger is the CAMP. And how is CAMP formed? By the activation of an enzyme, adenylate cyclase. Now, what happens is, the second Now what happens? The second messenger causes a change. How does it cause a change? It will now activate certain enzymes, all right? So here, as you can see, a protein kinase, which is inactive, is activated. And now once it is activated, it will then create a cascade effect. A substrate which is inactive, it will become active, and you'll see cellular changes. All right. So here, remember, prostaglandin affects the level of adenylate cyclase. Do you remember what we had talked about prostaglandin? We said prostaglandin helps to regulate hormones. How does it help to regulate hormones? Is it will just change the level of adenylate cyclase. If this level goes up, then what will happen to the amount of CAMP? Obviously, the amount of CAMP goes up. And if this goes up, then what happens? More and more of these other proteins or enzymes are activated, and the different substrates are activated. Okay. Here's an example. The example is epinephrine. Now, epinephrine is released in response to stress. All right. So epinephrine comes and binds here to the receptor. What happens? This is of course a liver cell. So what happens if this is stress? Remember, what do you want? You want more and more glucose, right? Okay. So in that case, what happens is same thing. CAMP is produced because adenylate cyclase is activated, and CAMP in turn will activate other enzymes that will break down glycogen into what? Glucose. So now the liver cell will produce lots and lots of glucose, and you're ready for stress. That is, you're ready to either flee or fight. All right? So many hormones use CAMP as a second messenger. All right? Now here what happens is, these are many hormones, of course we know that, that all the non-steroid hormones use CAMP as a second messenger. So this information is just repeated. But now we talk about another enzyme. All enzymes are going to be important. I'm putting a star mark. Phosphodiesterase. So this enzyme breaks down CAMP to make sure its action is short-lived. Now, why would this be necessary? Because always remember, go back to this slide, if you have CAMP, you are going to create a cascade effect and going to bring about that particular metabolism, activate that particular chemical changes, okay, those particular chemical changes. Now, if you don't have the second messenger, then the entire process will be stopped. All right. So this is analogous to the acetylcholine esterase. You should have picked that up in 201. You can review that. All right. So how the hormone levels are controlled in the body? But first thing, all the hormones, they have a certain amount of what? Half-life. All right. And this is so there's some hormones that act quickly and for a short time of time, or some have long-term effects. Now, go back and think about steroid and non-steroid hormones. Which one do you think will have a long-term effect? Of course, steroid hormones. They affect your what? DNA, all right, which is the blueprint of the cell. Now, hormone levels drop because of either they're broken down, typically broken down, or they're washed off in the urine. And remember what I said is you can increase or decrease secretion, and this is again to control hormone level. And in endocrine cells is required to control hormone level, and we will see this in just the next slide. So here, all hormone secretion is under negative feedback. This is something that you absolutely have to remember. All right. In fact, this entire semester long, we will learn only 2 positive feedbacks. Okay. And we talked about clotting, and the second one is birthing. Now, negative feedback control, remember, all hormones are controlled by negative feedback. How do we know that? in this you will see that the level of hormone in your body, in your blood, you will just see that it will never be extremely high. Why? Because there is a stability, and you can see this in the next slide. Look at the levels. So here you will see the average concentration remains the same. There'll be a slight increase and a slight decrease. As soon as there is a slight, this is what homeostasis is all about, right? So go back and tell me, tell me what is negative feedback. You should have picked that up in 201 chapter 1. So with negative feedback, what happens is that the product what it'll do is it will inhibit if the product accumulates. Okay, here I'll explain it better. A is being converted into B. All right, this is a chemical reaction. As soon as you have lots of B accumulation, B is going to inhibit its own release. All right, this is called negative feedback. So the product is going to stop or slow down the initial, will slow down the stimulus that actually produced that reaction. All right, so here, and as you can see, the hormone level, and this slide was just to show you the average concentration of the hormones. Now there are different ways, and we are going to talk about that as we pick up each hormone as we go along. All right, so yes, I think with this we come to end of our presentation for now. I thank you for listening to me. If there are questions or concerns, please come and see me. Thank you. Have a good day.